All right. Hello, everyone. My name is Kirill. I'm instructor from University of Texas Southwestern Medical Center, and today I would like to share with you several of my research projects. And my talk is entitled today "From Protein Domains to Cancer Subtypes: Harnessing the Power Classification Algorithms to Advance Biology." Classification is a approach that creates simplified models of the real world. And there can be various types of classification, like organisms, RNAs, or cancer subtypes. Today, I would like to share with you several projects that are belong to two different directions. First is classification of protein structure, and the second types. So let's start with the protein structures. Proteins consist of amino acids, and amino acids construct the secondary structure elements such as alpha helices and beta strands, which in their own turn are the building blocks for the protein domains. Domains are um, evolutionary, functional, and functional units of proteins. And as we heard today from the morning keynote lecture, that function structure intertwined, and it's especially true for the proteins and protein structures. And the classification of protein domains is especially important for understanding a better understanding the protein function, and especially function of related homologous proteins. There are currently exist several approaches, several databases, several classifications, and in our team developed and currently maintaining ECODE database. ECODE stands for Evolutional Classification of Protein Domains. And the main feature of this classification is the position of topology group below homology level, which allows us to detect real distant homology relationship between protein domains, which sometimes can be detected only manually. And with the arriving and popularization of artificial intelligence approaches, these methods became really popular and useful in many fields of research, and structural computation and biology is not an exception. I'm sure you have already heard about AlphaFold. It's a deep learning a method for identification of uh, 3D protein structure, and currently it's a very accurate method. And just last week, AlphaFold released a new version, and everyone is really excited to check it out and try it. And we also use this approach in our research. And the first project that I would like to share with you, which we use the AlphaFold in deep learning, is a study of so-called pan-cancer structurome. It's a structural space of over and underexpressed protein coding genes in 21 major cancer types from TCGA database. So we collected all over and underexpressed protein coding genes, and classify all the protein domains for known proteins, determined experimentally determined protein structures, and for those who don't have experimentally determined structures, we used AlphaFold models. And our analysis revealed, first of all, overrepresentation of beta sandwiches architecture in overexpressed protein coding genes. This architecture includes such proteins as immunoglobulins, interleukins, and galactins. These proteins are important element of inflammation, which very often is a important part of oncogenesis. And on the other hand, uh, we showed significant underrepresentation of exclusively alpha architecture. Uh, Proteins of this architecture are important elements of such processes of homeostasis, apoptosis, and transmembrane transport. So we believe that the cancer cell try to reduce representation of these proteins for successful oncogenesis. Another project was related to Salmonella enterica pangenome. Salmonella is a pathogenic bacteria which cause severe disease, and we used all available strains from NCBI database, Salmonella strains. It's about 80,000 strains, and extracted a non-redundant list of proteins and domains, and uh, using AlphaFold, we modeled all structures and classified all domains, and using genomic distance between. So for the we compared strains derived from human organism and environmental strains, and identified list of potentially pathogenic proteins that could contribute to bacterium virulence, and using um, genes distance between genes, we identified several novel potential pathogenicity islands that also uh, could contribute to of bacterium's virulence. And finally, one more uh, classification project. We using AlphaFold models, we classify domains of 84 whole proteomes of model organisms, and we believe this could be useful tool for the researchers who study these organisms. And now let's move on the second direction, is classification of cancer subtypes. So identification of cancer subtypes is important for better understanding and identification of the right treatment approach for a particular um, patient, and subtype specific therapy was successfully applied for various cancer types. And I've been working with the testicular germ cell tumor, um, and in particular seminoma type. It's the most common type of testicular germ cell tumor, and testicular germ cell tumor, and it's the most common cancer type in a young man between 14 and 40 years of age. Um, The current treatment of testicular germ cell tumor includes orchiectomy or surgical removal, radiotherapy, and platinum-based chemotherapy. Um, However, platinum-based chemotherapy can be responsible for more than 40 severe side effects. And moreover, about 20% of seminoma patients will relapse during the 1 year after the treatment, and the current reason for this is currently unknown. So, as a subtype-specific therapy showed its efficiency for some other cancer types, we decided to discover the possibility of identification of subtypes for seminoma patients as well. And we been studied the all available seminoma samples from TCGA database, and based on transcriptomic data. So first of all, I want to pay attention to the fact that seminoma tissue is highly infiltrated in lymphocytes, and so we removed all transcript related to immune cells, and to focus exclusively on the cancer cells, and we did the clustering, and clustering results showed two distinct clusters, which we call subtype 1 and subtype 2. and we studied the molecular features of these two subtypes. And first of all, over significantly overexpressed genes between two of these subtypes of pure seminoma revealed that subtype 1 include one gene which is signature for seminoma, which makes sense. However, subtype 2 include 11 genes which is signature for which overexpression is signature for non-seminomatous germ cell tumors, which are different type of testicular germ cell tumor and which are more aggressive and require different treatment approach. And long story short, we identified pluripotency markers and show that the rate of pluripotency is different between these two subtypes. Then we, based on overexpression levels, genes overexpression levels, we suggested that there are two different double-stranded DNA repair mechanisms are active in two subtypes. This is especially important because platinum-based chemotherapy mechanism is actually creating double-stranded DNA breaks. Then different loss of heterozygosity rate, and we also studied microenvironment in these two subtypes, and we hypothesized that potentially the failure of immunotherapy clinical trials for testicular germ cell tumors might be due to senescence of tumor microenvironment, and we identified some markers of senescence in seminoma subtype 2. So overall, the potential reason for relapse of seminoma patients we believe might be due to undertreatment of seminoma subtype 2. And moreover, we show that there might be potential transformation between seminomatous and non-seminomatous germ cell tumors, which can previously was also hypothesized by other researchers. And we also developed a deep learning decision-making tool based on histopathological slides available at the TCGA database to distinguish between these two subtypes using only images from these slides. And the most important thing here is that pathologists cannot distinguish between these two subtypes just looking at the slide. And that's why we try to deep learning approaches to help here. And the results show that the deep learning model showed that it was able to identify some morphological difference between these two subtypes. And we try to detect this particular morphological difference. And the first idea was to measure nuclear sizes between two subtypes. And however, the results show no significant difference between the two subtypes, which means there is something else yet to be discovered, and it's still work in progress here. So that's all I have for today, and I would like to thank all my collaborators and funding agencies, and thank you so much for your attention.
One quick question. Anybody has a question?
Thank you. I feel that annotation of domains is really gets much less attention than annotation of proteins. So I think your work is important.
Thank you.
Has there been in your work or anybody else's attention to the relationship between domain evolution and domain structure and subcellular localization?
That's an interesting question. We didn't study particular relation between subcellular localization, but yes, I'm sure that there is, for example, as you know, membrane proteins that tend to be alpha helical mostly. And
Could you get busy with it, please?
that's an interesting question. Yeah, thank you so much.